So hi everyone, I'm Shawn. I'm from Stephen Plotkin Lab from UBC Physics department, and I'm a PhD student. And this work is in collaboration with Promise Neuroscience company and our department. And alpha-synuclein is a intrinsically disordered protein that could misfold and aggregate it into Parkinson's disease patient's brain. So we want to develop an antibody treatment to clean out this alpha-synuclein and possibly treat Parkinson's disease. And our lab has previously developed an algorithm to predict misfolding prone region on alpha-synuclein we call epitope. And this epitope is cyclized and in terms of cyclopeptide, and this cyclopeptide is injected into the mice, and this step is called immunization. And from the sera of the mice, we select the best antibody that has property of oligomer selective ability. And this antibody is further validated for neutralization of some Parkinson's disease. And the whole study we focus on, in this research, we focus on the very first step on how to select the best cyclopeptide. So this is a purely computational work on to develop a virtual screening platform. And this work is will be submitted to JCTC journal very soon. You might have a question why oligomer selective property is important. Because among all the 3 species of alpha-synuclein, only the oligomer form is toxic. The monomer form is deemed as the healthy form, and so we don't want to clean it. And the fibril form is relatively inert than oligomer. And evidence has shown that targeting fibril could cause severe inflammation effect in the brain. And so we come back to the epitope. So the epitope we predicted is EKTK-EQ. And we cyclize this epitope using cysteine and glycine. So the figure on the right shows one of our cyclopeptide scaffolds. We call it 1,4-TKEQ because it has a sub-epitope region TKEQ and cyclized by 1 glycine on the left and 4 glycine on the right of the cysteine. Glycine is used because it is most chemically inert amino acid, so it could avoid causing unwanted immune response. And cysteine is used because it's needed to form a disulfide bond to a carrier protein like KLH in order to perform the immunization step. And by varying the number of glycine and sub-epitope region, we can generate tons of cyclopeptide like this. So a good cyclopeptide should have some criteria. It should be conformationally similar to oligomer, but not monomer or fibril. Since oligomer is the most pathologic species, so we want our immunogen to be as similar as possible to oligomer, but not other healthy species or not pathologic species. To compare this conformational similarity, we generate the conformational ensemble for each of the species, for each of the 4 species. And our lab have developed an algorithm to produce monomer ensemble, disordered monomer ensemble. This is done by random pivoting phi-psi angle. And for oligomer, we also have previously published paper on how to generate oligomer-like ensemble. This is done by partially unfolding the fibril structure. And it is achieved by forcing a collective variable Q, which is fraction of native contact, to only 65%. And so this oligomimicry ensemble is also called stress fiber ensemble. And then for fibril structure and cyclopeptide structure, we generate their conformational ensemble by normal molecular dynamics simulation, hundreds of normal molecular dynamics simulation. So we have the 4 ensembles in hand, then we can start calculating our conformational similarity. The conformational similarity is formalized into 2 metrics. The first is JSD, Jason-Shannon divergence. It represents how discernible are the 2 distributions. And the embedding depth is the other. It is a non-reciprocal measure where dA/B means the extent distribution A is assuming distribution B. 2 identical distributions will achieve the highest similarity in terms of JSD. On the other hand, for embedding depth, the highest similarity will be achieved if one distribution spike within the other. So if we compare our cyclopeptide ensemble with the other three ensemble species by these two metrics, we can generate 4 similarity measure. And they are listed on the y-axis on the first 2 rows of this diagram. And the x-axis just lists all the scaffolds. In total, there are 48 of them. Additionally, we sparse through our CSV protein data bank to calculate pathway target effect. And this reflects the fact that we want our antibody to only target oligomer, alpha-synuclein oligomer, but not any other random protein. So combining all these 7 criteria, we can put them into a small TOPSIS ranking server that our group also previously developed. And this server can generate a ranking. It can read the ranking from the table. Some of the top ranked scaffolds are further are patented and then further proceed to the immunization step. So we have the ranking in hand. To validate the ranking is a hard work, because that would involve testing the antibody, testing the efficacy of antibody that's raised by different cyclopeptides. However, to test the individual criteria, it's a more simpler job, because it could be done by some in vitro experiment. For example, we've done seeding activity assay to validate the 2 criteria, JSD, cyclic stress fibril, and depth cyclic stress fibril. Both of these 2 criteria stands for the similarity between cyclopeptide ensemble and the stressed fibril conformation ensemble. And the experiment, in the experiment, the monomer of the alpha-synuclein, the aggregation of the monomer alpha-synuclein is seeded by different constructs, and the y-axis marks the extent of the aggregation. And in the plot, we can clearly see that the cyclopeptide seed has more seeding activity than linear version of it. So this result again support the rationale using cyclopeptide as our immunogen. And we have done the same experiment for other cyclopeptides as well. And some preliminary result shows that the higher the similarity to stress fibril, there's a higher seeding activity. And the results are shown here, and it's still preliminary, but it will be available soon. And to sum up, in this research, we developed a virtual screening platform to select the best immunogen in order to proceed to immunization step. And the process starts by generation of ensembles, generation of fibril, monomer, oligomer, and lists of scaffolded epitope ensembles. And then we calculate the similarity measure between them. It's formalized by 2 measures, JSD and embedding depth. And then the ranking criteria are input into a small TOPSIS server, and then the top ranked scaffolds proceed to immunize mice and raise oligomer-selected antibody. And this platform in theory, can be expanded to any scaffolding method, not just limited to cyclopeptide, as long as the conformational ensemble could be sampled by simulation. The reason why we use cyclopeptide here is because it's a simple method to constrain the epitope diversity. And there are some other possible scaffolding methods that can achieve the same job. For example, we can stack mutable cyclopeptides laterally. This could mimic the environment of oligomer or fibril. Or another way to constrain the conformation diversity of the epitope is directly embed it in a folded state. And finally, we'll be happy to see if this platform are applied to other application. And thank you for listening.